Hello, can somebody confirm you can hear me?
We can hear you.
Great. Before we begin, are there any questions? All right, let me get my lecture up. Let me share my screen. All right, I had that we were talking about the physical methods for microbial control. Let me state that today is March 13th, and your final is coming up. I think it's next Wednesday. Oh, I've got too many papers here. That won't work. I am grading the infectious disease project. I hope I have it graded before you take your final. Figure about 10 minutes for each paper, and there's about 40 of them, so 40 hours worth of grading. Where am I looking? That's assuming I did the math right, of course. All right, your final will be Wednesday, March 19th. Please get it turned in by 11:59 p.m. After 11:59 p.m., you'll run out of time, and the exam, if you're taking it, like I said, will just close down. The exam will have heavy coverage of chapter 7, 13, 14, and lab module 9, because these are material that we've not been tested on yet, quizzed on yet. There will be questions from every chapter except for chapter 2. So there will be at least one question from each chapter, as well as from each lab. Any questions about the final? Remember, there are two parts. Part A, which are fill in the blank, short answer, and there's only a few questions in it, 3 or 4 questions. And then part B, which is multiple choice questions. We talked about the different ways of physical methods for controlling microbial growth, and filtration works by filtering out the microbes in the filter. You can also use low temperature to control microbial growth. Please understand, low temperature generally does not kill microbes. It just puts them in stasis. Obviously, they will die given enough time at low temperature, but it slows the metabolic rate of microbes so they cannot reproduce. Psychotrophs can reproduce at low temperature, down to about 0 degrees. And these are organisms that can grow in your refrigerator. They can reach numbers of 2 million starting from one cell within a week, which is why when you have stuff in the refrigerator, after it's a week old, you should probably throw it out. Unless there's preservatives in it, like salt or acids or lots of sugar, you should throw it out after about 2 weeks. Freezing does stop metabolic activity and does stop organisms from growing. It does kill, freezing and then thawing, I guess I should state, does kill all eukaryote parasites. So if you're ever wondering, geez, maybe this pork has some parasite in it, you can just freeze it and then thaw it out. That would kill all of the eukaryote parasites. Freezing does not kill, well, about half of bacteria. And I assume that's about half the bacteria cells will die with freezing and thawing, but about half of them will survive. It depends, I think, mainly on the formation of water crystals and whether the water crystals destroy the bacteria cell or not. But about half of bacteria can survive freezing and thawing. It does depend somewhat on the species. So if you wanted to remove the bacteria, freezing would not work. It would only reduce the bacteria numbers by about half. Freezing and thawing, I should state. Freezing does stop metabolic activity, so no microbe can grow once it freezes. Now, some microbes do put out, oh, what do they call it? A natural antifreeze. And so they can survive at temperatures less than 0 degrees Celsius. But if you get it cold enough, you can freeze the microbe even with it has antifreeze. Slow freezing in your freezer is what we're talking about, your home freezer. It allows ice crystals to form, which can kill some bacterial cells, about 50% of them, and all parasitic eukaryote species. Fast freezing, that's putting in liquid nitrogen, does not kill nearly any bacteria. And of course, the eukaryote parasites can survive fast freezing as well. Fast freezing or quick freezing is when you freeze things in liquid nitrogen. It freezes so quickly that the ice crystals do not have time to form. So it does not kill bacterial cells. Most people freeze things in their home freezer, in which case the eukaryotes would be killed. High pressure is another way that we can control microbial growth. High pressure transfers the pressure evenly throughout a liquid, killing vegetative bacteria cells, denaturing proteins, altering carbohydrates. It does not kill endospores. So endospores would not be killed by high pressure. A high pressure is sometimes used to preserve the color, the flavor, and the nutrient value, like of juices. And heating usually changes all of these, the color, the flavor, and the nutrient value. Like if you don't know, you don't want to boil anything if you want the vitamin C from it, because boiling will kill vitamin C. Any question about any of that? Like I said, some juices are pressure treated and then packaged and sealed, and that's how it uh, stays preserved. Desiccation is drying, and osmotic pressure can be used to control microbial growth. This prevents microbial metabolism. Cells can remain viable for years. If it's an endospore, it can be viable for, I don't know, at least 8,000 years because they've had endospores germinate out of the Egyptian mummies, which were in the desert, so they're very dry. Um, of course, this isn't desiccation, but they've had endospores surviving something like 40,000 years, or maybe it's 40 million years, a long number. And that's frozen in Antarctic ice, where they drill through the ice and take a core. They've had endospores germinate after a long, long time that way. That's not really desiccation, that's freezing. Examples of desiccation are dried fruit, dried figs, raisins, dried vegetables. Osmotic pressure is using high osmotic pressure from either high salt or high sugar or both, causing water to leave the bacteria cell, causing plasmolysis, or the plasmolysis is where the water leaves the cell and then the cell membrane shrinks away from the cell wall. Osmotic pressure can kill cells. Like E. coli is pretty sensitive to it. I'm not sure how quickly it would die, but about a day or less, E. coli would die under osmotic pressure. An example of a high osmotic pressure would be molasses. Molasses doesn't need to be refrigerated. You can keep it at room temperature because when microbes drop in the bottle of molasses, the water will leave the microbe going into the molasses, and that will kill the cells. Even an endospore, it will prevent it from growing or germinating. So high osmotic pressure is actually very effective. And we've been using osmotic pressure as well as desiccation for, gosh, I don't know, eons, millennia. People have been using desiccation and high osmotic pressure to preserve food. Examples of high osmotic pressure is molasses, honey, salted fish. Any question about any of that? All right, another physical method for controlling microbial growth is radiation. Radiation damages DNA. There's two types of radiation, ionizing radiation, which are X-rays, gamma rays, and electron beams. They have a short wavelength and high energy. That high energy, when it hits an electron, causes the electron to leave the atom, creating an ion with molecules of water or some other molecule. And then these ions can react with other molecules, especially with DNA, damaging the DNA. And you damage the DNA enough, you'll kill the microbe. Ionizing radiation is used for preserving food, pharmaceuticals, and medical supplies. You can sometimes find it in the grocery store, food that's been ionized and then packaged, and they just store it at room temperature because it's fine. There's no microbes in it. They can't grow and spoil the food. And the microbes have been removed by ionizing radiation. Some camping supplies also sterilized by ionizing radiation. The other form of radiation that damages UV light, damages microbes is UV light. UV light is a form of non-ionizing radiation. UV light causes adjacent thymines to form a dimer between them. Thymine is not normally bound to another thymine. This only happens when the thymines are adjacent to each other and exposed to UV light. These thymines will not pair with the complementary A in the second strand of DNA. This creates a bubble in the DNA and prevents the cell from transcribing this gene or replicating this region. So it can't transcribe it or replicate it without creating errors. Microwave radiation is not particularly antimicrobial, but if you use enough microwaves, it will heat up the microbes or the water around the microbes, which would then kill the microbes. Any question about any of that? All right. This table is showing you the summary of the different ways you can control microbial growth. See if I can blow this up. So we have moist heat, boiling, steam, and autoclaving. Pretty effective. Pasteurization is usually moist heat. I'm not aware of pasteurizing air, but I suppose you could pasteurize air. Dry heat, direct flame, incineration, very effective, turning the cells into char. Hot air sterilization or baking is not as efficient as moist heat. Filtration, another method of controlling microbial growth. Refrigeration or cooling things, stopping microbial metabolism. Deep freezing. We didn't talk about lyophilization, so I'm not going to talk about it. But deep freezing stops all metabolism. It does not necessarily kill the microbe, especially bacteria. High pressure, desiccation, osmotic pressure, all of these can stop microbial growth. And then radiation, both ionizing and non-ionizing radiation. All right, that's enough for the physical methods of controlling microbial growth. Let's talk a little bit about chemical methods of controlling microbial growth. Few chemical agents actually achieve sterility in controlling microbial growth. Most chemical agents simply decrease microbial numbers to safe levels or remove vegetative pathogens, vegetative cells. So chemicals are very good at being a disinfectant, but that doesn't sterilize the area, meaning most chemicals do not achieve sterility. They simply lower the numbers to safe levels, like when you disinfect your toilet. Disinfection is the destruction of vegetative pathogens. It reduces the numbers, does not remove all bacteria. There are some factors related to effective disinfection. The concentration of the disinfectant. Generally, the higher the concentration, the better. That's not always the case. Like when we're working with alcohol, 70% alcohol is better at killing than 95% alcohol. The time affects the disinfection. Generally, the longer you apply the disinfectant, the better it'll be. The pH affects the disinfection. Generally, the more acidic the environment, the better the disinfection. Although you could have the more alkaline too, meaning neutral pH will be the least effective. And then the presence of organic matter. For greater disinfection, you don't want organic matter around. The trouble is, organic matter is often found in clinical situations, and organic matter can neutralize or inactivate the disinfection. Like bleach, for example, is very easily inactivated by organic matter. There are different types of disinfectants, phenol and phenolics, compounds based on phenol, biphenyls, and other phenol. Although these are two, excuse me, two that are hooked together. Biguanides, halogens. I'll talk briefly about halogens. This would be chlorine bleach and tincture of iodine or halogens. Alcohol, rubbing alcohol, an example of an alcohol. Heavy metals like copper or what's the one that, well, I guess silver can be applied to a patient's like dressing or wounds to prevent and remove microbes, kill microbes. Surface active agents called surfactants are agents frequently used in hospitals nowadays because they're very good at being a disinfectant. There are chemical food preservatives. We'll talk just slightly about that. Antibiotics aldehydes, like formaldehyde, gaseous chemosterilizers. Oh, I forgot the name of that. Well, ozone would be, well, I don't know if ozone is actually gas, but uh, chlorine gas would be a gaseous chemosterilizer. And then peroxygens like hydrogen peroxide and oxidizing agents. Few of these disinfectants kill an endospore, but actually the peroxygens like hydrogen peroxide do kill endospores. I think the other one might be aldehydes. Most of them do not kill endospores. Like bleach kills only about 50% of endospores. Sorry about this, kind of blow my nose here. Um, I think bleach kills certain species of endospores, but not other species. When we talk a little bit about this table, you should focus on the first three columns of this table. So I'm actually hiding the fourth. That's not when whoop, I guess I'm not. I'm supposed to be hiding it by putting that over there. There we go. Uh, phenolics, biphenyls, the mechanism of action, you don't really need to know that. The preferred use is actually as a disinfectant. Biguanides, like chlorhexidine, also skin disinfection, surgical scrubs. Halogens, I did mention this once before, chlorine bleach and tincture of iodine. Iodine's a fairly effective antiseptic. Used to be used all the time in hospitals before surgery, but they've come up with better agents now, so it's not used as frequently as it used to be. Chlorine glass and bleach, halogens, very effective disinfectants, but remember, uh, they are inactivated by organic matter. Alcohols, very poor disinfectants. We usually call them degerming agents because they just reduce the number of microbes to about safe levels, but they do not clean the surface. There are still microbes on your skin after you alcohol swab it, but it reduces it to safe numbers for the needle to puncture the skin. Heavy metals, for many years, they were not used by the medical profession because better agents became available, and these agents are toxic to humans. However, because of the development of antibiotic drug resistance, as well as other resistances that microbes have been developing, their use has come back. Silver nitrate is sometimes used to prevent gonorrhea uh, being transmitted to a neonate, meaning a baby that's about to be born. Uh, Copper sulfite used as an algicide. Like I said, heavy metals have come back to use in medical treatment, especially with what do they call it? Band-aids and gauze to prevent microbes from infecting the wound. And their use has come back largely because microbes have become resistant to many forms of antibiotics. Surface active agents include soaps and detergents, sanitizers, quaternium ammonium compounds. They're used obviously in the house, soaps and detergents and sanitizers. Quaternium ammonium compounds used more in the medical industry. It's an antiseptic for skin, so it's very widely used when treating skin, as well as it's used in the hospitals and medical establishment for cleaning utensils or goods that would be damaged by either heat or another agent, like surgical instruments, maybe. Although usually surgical instruments can be autoclaved, which would be better than a quaternary ammonium compound. But if not, you would use a quaternary ammonium compound. Chemical food preservatives include organic acids, nitrates, and nitrites, which are added to food to keep the food from spoiling. Meat products like ham, bacon, hot dogs, sausage have nitrates and nitrites in them. And that allows the agent, the food agent, to last longer than it would if it didn't have the agent in it. Now, obviously, a hot dog can still go bad, but you can keep it in the refrigerator for probably over a week because of the nitrates and the nitrites. Any question about any of that? Calcium propionate is an organic acid which is used in bread to help preserve the bread. If you know, when you buy store-bought bread, it can stay generally for about 2 weeks at room temperature before mold will start to grow in it. And it's because of the food preservatives. If you didn't have the food preservatives in it, it could only stay for about a week. Aldehydes, like glutaraldehyde, formaldehyde, used as disinfecting, like used in medical equipment. Chemical sterilizers, the best one being ethylene oxide, that's the name of it, I couldn't remember, is a gas. It actually is lethal to endospores, although you need long-term exposure. You can sterilize things with ethylene oxide that would be damaged with other agents. I remember when the anthrax spores were going through the mail in the United States. Somebody was mailing anthrax spores to people they were actually disagreeing with politically, and they treated the contaminated envelopes with ethylene oxide. And then peroxygens, I did mention this, like hydrogen peroxide. It's used as a antiseptic on wounds, for example. It can also be used as a disinfectant, and it is very effective at killing endospores. The reason is endospores are produced by bacteria which are sensitive to oxygen, and hydrogen peroxide can kill those organisms. Any question about any of that? All right, comparing the effectiveness of various antiseptics. You can see here that soap and water is not very effective at killing microbes. At 1 minute of washing your hands in soap and water, you're killing less than 10%. And most people don't wash their hands for 1 minute. Tincture of iodine is much better. And after 1 minute exposure to tincture of iodine, you kill about 80% of microbes. You get about 90% of microbes after 90 seconds, meaning a minute and a half. So much more effective than soap and water. And the nice thing about tincture of iodine is once you apply it to the skin, it remains active for a long period of time. And that's why it was used in surgery, because you applied it to the skin, it wasn't irritating to the skin, and then it could stay on the skin for long periods of time. And you only need 90 seconds to kill 90% of the microbes. Let's see, what would that be? Would be, but it doesn't matter. Oops. All right, let's talk about some of the microbial characteristics that affect microbial control. There's actually a hierarchy of the relative resistance of the major microbial groups to chemical biocides. This is a simplified hierarchy of the relative resistance, and that is prions are the most resistant microbe to any chemical treatment. Essentially, there is no chemical treatment that will kill or inactivate prions. There could be one exception. We'll talk about that later. But generally, prions are very difficult to kill, and essentially no chemical agent can kill them or inactivate them. Endospores are the next most difficult thing to kill. Most agents do not kill endospore. I did mention gaseous agents like ethylene oxide can, but you need long-term exposure. You need over 20 minutes to kill endospores. Obviously, hydrogen peroxide can kill endospores. So if you want to kill endospores, you use a peroxygen or some other agent that can kill endospores. Mycobacteria, the next most resistant. Mycobacteria has a lot of mycolic acid in their cell wall, and this tends to keep the cell from drying out. And so it keeps the cells alive, but it also keeps anything that can dissolve in water outside of the cell. And so any aqueous agent is not going to be good at killing mycobacteria. Any question about any of that? All right, cystic protozoa, the next vegetative protozoa, gram-negative bacteria, the next item. The gram-negative bacteria are more resistant than the gram-positive bacteria because the gram-negative bacteria have an outer membrane on their cell wall, which prevents a number of things from gaining entry into the cell and then killing the cell. Any questions about that? The gram-positive bacteria, the agent can get right through it and then kill the cell. Obviously, the best example would be penicillin G. It cannot get through the outer membrane of the gram-negative cell, but it can go right through the gram-positive cell wall and then kill the cell. There are some detergents that work this way as well, and other antibiotics and other agents. Just please don't ask me because I don't know the other agents off the top of my head. I used to know the name of another family of antibiotics that did not kill gram negatives, but I would have to look it up now. I don't remember. It's a family of antibiotics that's similar to the family penicillin, has the same active agent, but it's a different family member, meaning it has a different biochemical structure than the penicillin family. Then fungi, viruses without envelopes are a little more easy to kill, or a little more, excuse me, a little harder to kill than viruses with the lipid envelope. And then we have gram-positive bacteria, one of the easiest things to kill. And then the easiest thing to kill or inactivate would be viruses with lipid envelopes. That lipid envelope is sensitive to drying out, it's sensitive to chemical agents, and it may be more sensitive to the protein coat. And that's why viruses with lipid envelopes are easier to kill than a virus without an envelope. Like if you don't know, the viruses that cause the human cold, which are naked viruses, not all are them, but some of them are, they can survive much longer and better than a virus with a lipid envelope, even if the virus is causing the same disease, the human cold. Like coronaviruses, they're more sensitive to dying out or being inactivated than a virus that causes the human cold that does not have an envelope, like the rhinoviruses. Any question about any of that? All right, I've talked about that already. The porins being the pore or the membrane channel protein in the outer membrane of the gram-negative bacteria. All right, prions. How do we control prions? Prion is a proteinaceous infectious agent which forms spongiform encephalopathy. Most prions are resistant, I should say, most uh, chemical agents are ineffective at treating prions. How do we kill prions or inactivate them? It's not completely solved yet. You can incinerate them, raising them to 1,000 degrees, either Fahrenheit or Celsius, I don't remember, but it's 1,000 degrees to incinerate, and that does kill the prion. It's very unusual that a protein requires 1,000 degrees to kill the protein. Most proteins are inactivated simply by boiling temperatures. But a prion, there's something about it that is much more resistant to, well, heat, for example. They're resistant to everything but heat. There is one agent which they do use in sterilizing not chemical, sterilizing surgical instruments with to remove prions. And that is you can use proteinases. Treat the surgical instruments with the proteinase, and that destroys the prions. Beyond that, and then incineration, uh, those are the only treatments we have to remove prions. If you remember, the uh, British cows came down with mad cow disease. What they did was they incinerated their entire herd of cattle. All right, let me go back one step. Uh, and then mycobacteria, I mentioned this before, that it is very effective at removing or preventing the chemical agent that's water soluble from gaining entry into the cell and then killing the cell. Like bleach is not very effective against mycobacteria because bleach is water soluble. It doesn't get through the mycolic acid. However, mycobacteria are very sensitive to alcohols. Alcohol gets right through the mycolic acid, and then the alcohol can destroy the mycobacteria. Remember, alcohol is an agent that is a very poor antiseptic. It's so poor we don't even call it an antiseptic. Usually we call it a degerming agent. It removes microbes from the area that's been swabbed, but it doesn't really clean the area. All it does is de-germ the area, reducing the microbes to safe levels. However, like I said, mycobacteria are very sensitive to alcohol because it can get right through the cell wall and then damage the cell. So this is one characteristic of mycobacteria that doesn't fit the normal hierarchy. Mycobacteria are very sensitive to alcohol. Any question about any of that? So while microbial control methods are effective, especially the biocides, they are not uniformly effective against all microbes. The compounds discussed in this lecture are not generally useful in disease treatment of a patient, except for the antibiotics that we talked about. And most of the antibiotics I was talking about were antibiotics that they add to food to help preserve the food. Like they add an antibiotic to cheese and bread to prevent the cheese and the bread from spoiling. And obviously, those antibiotics are safe to eat. Any question about any of that? All right, that's it for chapter 7. Any questions about chapter 7? Let me briefly state the main points of chapter 7. Obviously, know the terms. Be able to discuss the factors that influence microbial death rates. Be able to discuss the three main ways in which antimicrobial agents kill or inhibit cells. 1, they damage DNA. 2, they damage proteins, like inactivate proteins or cause the protein to unfold. And then 3, they damage cell membranes or other membranes. And then 4, understand the various types of disinfectants and how they work. And then 5, understand the relative resistance of the major microbial groups to chemical disinfectants. So the last point is right here. You should know prions are the most resistant, endospores the next most resistant, mycobacteria the next most resistant, everything else less resistant, gram-negative bacteria being more resistant than gram-positive bacteria, and then the agent or the microbe most sensitive to any chemical agent is the viruses with a lipid envelope. Any question about any of that? All right, that's the review for chapter 7. Let me go to chapter 14. I'm just going to go by chapter number. If you're not wanting the review, you can leave this lesson. If you want the review, please stay and I will continue to give a review. Know the terms of chapter 14. If you know the terms, you'll know about 50% of this chapter. You should also understand the relationship between the normal microbiota and its host. All people, all animals, and all plants have microbes living with them in symbiosis, and this is the normal microbiota. And that happens shortly after birth, or during birth, actually, some of them. You should understand the different stages of disease development. Let me come to that. While I'm here, you should understand Koch's postulates. We've talked about Koch's postulates 3 times. The first time was actually in chapter 1, and I don't remember the second time. And then obviously in this chapter, chapter 14, we talked about Koch's postulates. So you should know that it was used to prove that a given microbe causes a given infectious disease. I guess you should know that there are, in the viruses affecting animals, there are a lytic animal virus, a budding animal virus, a latent virus, and then um, oh, what's the name of it? Persistent viral infection uh, virus. So there are those different life cycles. The latent life cycle of a virus usually has another life cycle, which is usually the budding life cycle, and it can leave the budding life cycle and then go dormant. The virus goes dormant, entering the latent life cycle of the virus. The virus can come out of latency. Usually, a stressor will trigger the virus to come out of latency and then re-enter the budding life cycle where the virus can replicate and go through metabolism and then cause disease. You should know the patterns of disease development, the predisposing factors that can cause someone to be more likely to get disease. You should know the development of disease. You should know that all diseases start with an incubation period, which is when the patient first becomes infected with the microbe, till the patient has any signs or symptoms. Some individuals only have incubation period, meaning their disease will be asymptomatic. Well, I guess they have incubation period and period of convalescence. They never have anything in between. The always asymptomatic. Uh, Most diseases have a prodromal period that follows the incubation period. This is when the patient first has signs or symptoms of disease, and the signs or symptoms are mild. This followed by the period of illness, when the signs or symptoms of disease are no longer mild, they're severe. That's followed by the period of decline, which happens when the patient starts feeling better, like when a patient has a fever and then the fever breaks, that's starting the period of decline. Generally, if you have a cold and then you start feeling better, that's the period of decline. Uh, the period of convalescence is when the number of microbes inside you drop below a threshold number and they no longer cause any signs or symptoms of disease. You still have the microbes, so you're still either contagious or communicable. There's the word I'm trying to get to. Communicable, because you still have the agents, but there are low enough numbers they no longer cause any signs or symptoms of disease. And that will, of course, end with the patient being cured. All diseases go through these periods, although not all diseases have all of the periods. Like I mentioned, the flu sometimes never has the prodromal period. It goes from the incubation period to the period of illness. And then some diseases never have the period of decline and period of convalescence, particularly if the disease kills the patient. And then some asymptomatic diseases, as I mentioned, only have the incubation period and the period of convalescence. Any question about the periods of disease development? You know about reservoirs, the transmission of disease. You should know about nosocomial infections. Nosocomial infections are one of the most important topics you can learn in microbiology. Because if you work in the healthcare industry, you need to know about nosocomial infections and how to prevent them from happening. All right, any questions about chapter 14? Let's move on to chapter 13. All right, so know the terms of this chapter, understand the characteristics of viruses, their structure, their classification. Remember, viruses are classified by a common English name. They do not have any Latin names. You should understand about viral multiplication and then the life cycles where the virus multiplies. With bacteriophages, these are viruses that infect bacteria cells. They only have two life cycles. The lytic life cycle, will the virus infects the host cell, replicates, and then lyses the cell, releasing the newly replicated virions. And the other life cycle is the lysogenic life cycle, where the virus DNA integrates into the host cell chromosome and then lives in harmony with the host cell. The trouble is, once the lysogenic life cycle ends, the lytic life cycle can begin, and then the virus can replicate and then lyse the cell to release the newly replicated virions. Let's see, I mentioned the lysogenic virus will integrate into the host cell's chromosome. Oh, and to leave the lysogenic life cycle, the viral DNA will be excised from the chromosome and then enter the lytic life cycle. Any question about any of that? All right, understand about prions and viroids. You should know that a virus' main objective is just to replicate, and they are inert, essentially have no metabolic activity outside of their host cell. They require a living host cell in order to live, using the host cell's enzymes, nucleic acid, amino acids, ATP. Viruses have either DNA or RNA for their genome, never both, either one or the other. And then a protein coat surrounding the genome. All viruses have these two things. Some viruses have a lipid envelope outside of the protein coat, but not all viruses do. The host range is the range of cells that can be infected by the virus. Most viruses only infect the cells of one or two species. HIV is a good example, infecting only the T cells, macrophages, brain cells, a few other cells of humans and of one of the chimpanzees. It does not infect any other species, and generally it does not infect other cells, like it doesn't infect your skin cell or your nose cell or your bone cells. All right, you should know that nucleic acid can be DNA or RNA, and if it's DNA, it can be double-strand DNA or single-strand DNA. The eukaryote cell has both of these. Our DNA is normally double-stranded, but when we're transcribing genes and when we're replicating DNA, our DNA, at least a portion of it, can become single-stranded. The genome can also be double-stranded RNA. That's unheard of in our cells. And then the viral genome can be single-stranded. And if it's single-stranded, you need to state whether it's the positive single-strand RNA or it's the negative single-strand RNA. Any question about any of that? Should know the general morphology of viruses. Or you should also know that the viral infection, when it infects an animal, can stimulate the animal to produce antibodies. These antibodies bind to and recognize specific cell surface molecules on the infective microbe. And these furthest out molecules, besides being antigens, they can also be important units within the virus, such as it might be the ligand, which attaches to the host cell receptor. And it can be other important molecules too, like coagulase and Staphylococcus aureus. All right, you know the viral taxonomy. Viruses end, the family ends in the name Viridae. The genus names in the name virus. So for the herpes family of viruses, we'd call it herpes viridae. And then for the genus, herpes virus, or maybe it has a more specific name. If there's more than one genus in the family, herpes viridae. And then there would be the virus species sharing the DNA as well as the general atmosphere or environment that the cell is living in. And then below the viral species, we have the subspecies. It'll be designated by a number, like there's sorry, I'm not getting an example here. Poliovirus, there we go. Uh, poliovirus type 1 is very similar in the same species as poliovirus type 2. They only have 6 mutations between them, but poliovirus type 2, the subspecies 2, is what we use for the live oral vaccine to prevent the patient from getting polio, the disease, as well as the microbes causing it. And then subspecies type 1 is the poliovirus, which causes the disease polio. Any question about any of that? I already talked about the lytic life cycle and the lysogenic life cycle for bacteriophages. And then for animal viruses, I've already talked about uh, the 2 phases you must know are the lytic life cycle of an animal virus and the budding life cycle of an animal virus. But there are 2 other life cycles, the lytic life cycle and the persistent infection life cycle. There are some consequences of lysogeny. The main one is that for the cell that's infected, actually there's 2 main ones, I guess. And if this is infected with a lysogenic virus, another lysogenic virus of the same species cannot infect this cell. And then the species has new DNA in it. This new DNA from the virus can code for something which will change the phenotype of the host cell. The best example is Corynebacterium diphtherium. The toxin is made only when the bacteria has a lysogenic virus inside its chromosome, because that Corynebacterium diphtherium toxin is encoded by the virus DNA. Any question about any of that? talked about most of these. You don't need to know the DNA family of viruses or the RNA family of viruses. With the exception, you need to know the family if we talked about it. But if we didn't, I won't be asking any questions on it. And the same with the RNA family of viruses. You should only know the families that we've talked about. Like Coronaviridae, we did talk about it. That's the number one cause of the human colds. It also causes polio. There we go. I knew there was something else. The Coronaviridae causes. The Coronaviridae causes polio and the human colds. It causes other diseases as well, but those are the two I talked about. Herpes viridae causing things like chickenpox, shingles. Okay, that's Hepaviridae. Sorry. Let me blow it up so I can read it. You don't need to know about Herpes viridae or any of these. You should know about Coronaviridae and Retroviridae because I did talk about those. You should know that Orthomyxoviridae is the virus that causes the flu. And this virus is different. It's similar to Coronaviridae, I guess. They're different in that they tend to change and mutate very rapidly. And that's why the vaccine cannot last a long time. And every year they actually change the vaccine depending on what virus species are spreading at the moment. These species do change rapidly, and that's why you need to get a vaccine at least every 3 years, if not sooner. But not all viruses are that way. A large number of them do not change rapidly, like the polio virus. Yep, I'm not doing the right thing here. Uh, the vaccine, which we made in the 1950s, both of them are still effective today. And that would be one of the Picornaviridae viruses. All right, any question about the RNA family of viruses? About Retroviridae, it has reverse transcriptase coded in its own genome. There's also a protein of reverse transcriptase in the virus. And that way, when the virus infects a cell, the reverse transcriptase can directly and very quickly make single strand RNA from the DNA and then use that single strand RNA to make double strand DNA, make single strand DNA and then use the reverse transcriptase to make double strand DNA. And then that double strand DNA can integrate into the host cell chromosome, essentially merging with the DNA or fixing with the DNA, essentially doing that without any chemical tools or not chemical, physical tools like oh, didn't mean to do that. Now, the coronaviruses cause one, the human cold, two, COVID-19, three, SARS, and then 5 MERS. SARS, MERS, and COVID-19 are all related to each other, and they are also more lethal than the flu, with MERS being the most lethal one, being the lethality of humans being around 36%. But fortunately, MERS does not spread from person to person. It tends to spread from camel to person, and you have to be in close contact with the camel for a long period of time to get MERS. All right, any questions about chapter 13? Let me state that this is showing you an infection, the number of infective virions on the y-axis and the time on the x-axis. An acute infection like the cold, it's very rapidly, develops very high initially, and then falls very rapidly. So it's gone after a fairly rapid time, about a week. That differs from a latent infection, where the latent infection first has the budding life cycle, like when the child first gets chickenpox, they're in the budding life cycle. Then the virus can go latent. This latency can last for years, and then the virus will start replicating, causing shingles in the patient. The first time the virus grows in a patient, it causes chickenpox. The next time it grows only in certain nerves, and we say the virus is localized to that one nerve. Anyways, that's the latent infection, where you have 2 humps, the second being smaller than the first. In between, you have nothing. The microbe's still in you, but it's not causing disease, and it's not replicating either. And then there's a persistent viral infection. It starts generally slow, mild symptoms at first, gets progressively worse with time. And then 3 things can happen. First, the virus can stabilize, the host can, what's the term I'm looking for, the virus can stabilize with the host and establish a balance, and the virus load stays about constant. And this patient will tolerate the virus. You can also have a persistent viral infection, which climbs very rapidly in the second latent infection, and then can climb down also rapidly, but it doesn't have to be. More likely than that, if it is going to climb very high, you get a bloom of the virus, and the patient will likely die. HIV tended to follow this life cycle, where you would have a persistent infection which would climb very slowly over time, and then it would bloom, and then the patient would die. The blooming may not be related to the dying, although it could be. It could be that the number of HIV cells in the body just became too numerous. But AIDS patients are most likely going to die from the lack of the immune system, in which case HIV is not directly killing the patient. It's killing the immune cells, but not the patient. Any question about any of that? All right, you should know about prions and viroids. Go to chapter 8. You should know a few points of chapter 8. You have been tested on chapter 8, so there'll be fewer questions, maybe 2 or 3 on the final. Oh, where is it? There it is. So once again, know the terms. You should know what an operator is, what a promoter is. excuse me, you should know about transformation, conjugation, and transduction. You should also know how the DNA replicate, how the DNA transcribes in the messenger RNA, and then how the messenger RNA is translated into proteins. And then lastly, you should understand the different types of mutation, like what's the silent mutation, what is a frame shift. Any questions about chapter 8? I'm assuming you're going to know the terms, so I'm not going to go through the terms. You should know that the strand, a double-stranded DNA is anti-parallel, where one strand faces this way, the other strand faces the opposite direction. And the DNA is held together by the sugar phosphate backbone, where sugar touches the phosphate, which sugar touches the sugar, which touches the phosphate, etc. You should know about DNA. You know about the central theory and central dogma or theory in microbiology. That is, all living cells come from a pre-existing cell. All right, we already talked about that, so I'm going to skip it. All right, any question about chapter 13? All right, you don't need to memorize this genetic code, but you should realize that there is a genetic code, and sometimes the same amino acid can be coded by more than one codon. And that's because there's only about 20 different amino acids in all species, the highest being 23, a microbe that has 23 amino acids, but they don't have to be the same amino acid as humans have. All right, any question about any of that?
Are you able to upload these PowerPoints onto Canvas?
Yeah, I will do that right after I'm finished here. You can remind me before I log off. But I meant to do that. I'm not sure which ones are up and which are not. Is chapter up 8 up? Chapter 8 up? All right, you should probably know about the complications in uh, protein translation and RNA transcription. And that is, in a prokaryote cell, you can have more than one RNA being made from the same gene at the same time. Obviously, this one started before all these others, but the point is you can have multiple RNA being made at the same time. And in prokaryotes, you can have the protein translation begin before RNA transcription finishes. In eukaryotes, you don't have the transcription and translation complication because transcription occurs in the nucleus, translation in the cytoplasm. However, you do have another complication, and that is in a eukaryote, in the DNA, the gene, there will be regions of the gene that code for the protein, and that we call an exon. And then there are regions of the DNA of the gene that do not code for the protein or do not code for the messenger or the RNA if it's only making RNA. So they do not code for the gene product. And that we call an intron, region of the gene that does not code for the end product. the introns have to be removed from the initial RNA transcript, and then the exons coded, brought back together, spliced together. And then this final messenger RNA now can leave the nucleus. The pores control what messenger RNAs can leave and what cannot. And if it has an intron in it, it cannot leave the nucleus. All right, I already mentioned you should know about the operon model of gene expression. You should know about mutations. All right, we talked about this too. All right, let's skip on this and go to the next chapter. Chapter 7. We already did that one. Let's move on. Chapter 6. Chapter 6 will only have 1 or 2 questions on the final. So you should know the terms, understand the various requirements the cells have for growth, understand the 4 phases of growth, and compare the different methods we have for measuring microbial growth. Let's talk a little bit more about the phases of growth. You should know about the requirements the cells have for growth. You should know the 4 toxic forms of oxygen. You should know the different types of culture media. Okay, here we go. The bacterial growth curve. Start with the lag phase. We then have the log phase or exponential phase. There's followed by the stationary phase, which is followed by the death or decline phase. And you should know during the lag phase, the microbes are not growing. They're making the enzymes they need to grow in their new environment. You should know why the exponential phase begins and why it starts to end. You should know why the stationary phase occurs, and then know why the death or decline phase begins. Any question about any of that? All right, that's it for chapter 6. Let's move on to chapter 5. Particularly what are photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. Know about, understand what aerobic respiration is, its steps, and how it differs from anaerobic respiration. You should also know what fermentation is. We did briefly talk about that. And understand what feedback inhibition is. You should know the 3 mechanisms cells have for making ATP, substrate level phosphorylation, where you just take phosphate from one molecule and add it to ADP to make ATP. You can also make ATP with respiration. This is where a molecule has high energy, like glucose, and the electrons are taken from glucose and put through an electron transport chain where one molecule carries the electron to one molecule, and then that molecule is oxidized and reduced, meaning this molecule gives the electron to the next molecule, giving it to the next, 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 till you get the last molecule accepting the electron. This is called the final electron acceptor. In aerobic respiration, the final electron acceptor is oxygen. In anaerobic respiration, the electron acceptor is some other molecule other than oxygen, something like nitrite or nitrate or sulfate. There are many different types of anaerobic respiration, and they each have a different final electron acceptor. Like nitrate, anaerobic respiration will have nitrate as the final electron acceptor. Any question about any of that? I guess you should know about photophosphorylation being the third way that cells can make ATP. And this only happens in photosynthetic cells. You should know the different steps of aerobic respiration, 3 steps, and then what happens in each of the steps. Mainly you make ATP. Anaerobic respiration has the same steps as aerobic respiration. The only difference is that the electron carrier chain, let's see if I can find that now. There we go. The electron carrier chain is smaller. There may be less proteins in this chain, so it makes less ATP. And the other difference is obviously the electron does not flow to oxygen. It flows to a different molecule other than oxygen. Should know about fermentation, lipid and protein metabolism. And then you should know about photosynthesis. There are 2 photosynthetic reactions where the cell makes a product from light. And one is cyclic photosynthesis. The other is non-cyclic photosynthesis. You need to know non-cyclic photosynthesis is the photosynthesis you probably were taught in your earlier biology class, where you start with 6 carbon dioxide, 12 water molecules in the presence of light. You make 1 glucose molecule, C6H12O6, 6 oxygen, and then 6 water. The other photosynthetic reaction is a cyclic photosynthetic reaction, and you only make ATP. The light is used to strip electrons from water, and then those water electrons are used to make ATP in an electron transport chain. All right, any questions about chapter 6? Chapter 5, sorry. Let's move on. We have got a few more chapters here. Sorry, I'm going over. Know the terms of chapter 4. You should know how the gram-positive and gram-negative cell walls differ, and describe the importance of taxes. You know the different types of cells and the cellular growth arrangements. Then you should know the different structures of prokaryote cells. There is no question about the structure of a eukaryote cell on the final. You should know about gram-positive and gram-negative cell walls. You should know that there are some atypical cell walls, the mycoplasmas. These are bacteria cells that lack a cell wall. And then the mycobacterium, There are also bacteria that are atypical, and that's because they have mycolic acid in their cell wall. And then the archaea are a different domain, and they have a different cell wall from bacteria. About the different ways cells can move things across their membrane. There's obviously a passive process using a channel protein, and then active transport using a protein pump to pump the molecule across the cell membrane. And this is called active transport. It does require ATP, one ATP for each molecule moved across the membrane. Prokaryotes also have another form of active transport. It's called group translocation, only found in prokaryotic cells, where the molecule that is moved into the cell is chemically altered. Like glucose, if it moves into the cell by group translocation, it is altered to glucose 6-phosphate. All right, you should know about endospores. We've talked about that topic 3 different times in this class. The first time was chapter 1. The second time was chapter 4. And I don't remember the third time. And then the last thing to know is the endosymbiotic theory. Any questions about chapter 4? Chapter 3, I believe, is the Gram stain. You should know the Gram stain. You should know about the sizes of organisms under the microscope. You should know why staining improves resolution. Staining only does one thing, and that is it either stains or changes the contrast between the background of the cell and the cell itself. By either staining the cell or by staining the background. You don't have to stain the cell, you can stain the background of the slide. That's a negative stain or a whatever. A negative stain stains the background. You should know there's 3 different types of stains. There's a simple stain, a differential stain, and a special stain. A special stain is a stain that stains something special inside the microbe, meaning it could be staining like the flagella, could be staining the capsule. That's a special stain, one specific structure of the cell. And then understand the Gram stain, its procedure, and the basis for its differential staining. Any questions about chapter 3? All right. For those of you who got through the review, I'll tell you there's no questions on chapter 2 on the final. You do need to understand chemistry to answer some other questions, but there's no question about chapter 2 on the final. On chapter 1, there are questions on chapter 1. Chapter 1 was an introductory chapter where we discussed a lot of topics. For the first time in chapter 1, like we first talked about the cell wall, we first talked about endospores, we first talked about Koch's postulates in chapter 1. And then we talked about each of these topics about 3 different times throughout the term. And so there will be a question on each of those 3 topics. And we first talked about them in chapter 1. So there will be more questions on chapter 1 than other chapters that we've already been quizzed on just because of the nature of chapter 1. And then chapter 1 also has a brief history of microbiology. Do I like the history of microbiology? Can anyone answer that? All right, I'll answer it since nobody's answering it. Yes, I like the history of microbiology. And so I tend to ask about 3 or 4 questions on the history of microbiology. To know the history of microbiology, you should know the major events in the history of microbiology and who discovered them. Except for the very first history of microbiology, talking about when we first saw fossils of living organisms on the planet Earth, you do not need to know any dates for this class. So you don't need to know any dates for the history of microbiology. All right, any questions on this chapter? If not, that's it for the review. Good luck on the final. Any questions before I go?
Yeah, are we gonna have lecture on Tuesday?
No, next week is finals week, isn't it?
So no more lectures?
Yes, this is the last lecture.
Okay, thank you.
All right, and the last lab, obviously. All right, bye,
Thank you. Bye.
bye.